Our paper focuses on the HIV protein VIF, which hijacks the host ubiquitination complex, ubiquitinating the antiviral ApoBec protein, leading to its degradation. We were interested in the dynamics of the HIV VIF ubiquitination complex. We ran molecular dynamics simulations of the VIF VCBC complex and the VCBC complex bound to the scaffold protein COL5. In figure 4 of our paper, we characterize the dynamics of the complex using principal component analysis to extract the principal components of motion. As you can see, the VCBC complex samples more alternate conformational states than the VCBC complex bound to COL5. Focusing on individual structures in the VCBC complex, cluster 1 is the most similar to the VCBC COL5 crystal structure indicated by a black X. Additionally, almost the entire VCBC COL5 ensemble falls into cluster 1. In cluster 2, the complex is more closed, and it is also twisted so that CBF beta in green and the VIF alpha beta domain move backwards relative to cluster 1. In cluster 3, the clamshell is closed even more, and CBF beta and the alpha beta domain of VIF are now twisted in the opposite direction, moving forward. Importantly, in the VCBC COL5 ensemble, we identified the same types of clamshell opening and closing and twisting motions of the complex. However, the sampling along these types of motion is reduced, as indicated by the magenta and gray distances on the figure, which have not changed as much in the VCBC COL5 ensemble as they do in the VCBC ensemble. Overall, figure 4 allows us to examine the alternate conformations of the VCBC complex that we find in our molecular dynamics simulations, and that could be important for HIV VIF function, as well as for identifying off-pathway conformational states that might be taken advantage of for disrupting HIV VIF function.